Hey dear students, welcome to my YouTube channel. So in today's video we will study about the bioluminescence. So I have two books. We will follow both of these. First we have an introduction to fish. So this book of S.S. Khanna, and secondly we will follow the Pandey and Shukla book. Okay, so mostly we will follow this book, that is the S.S. Khanna, but some topics like mechanism and some examples we will add from Pandey and Shukla book. So firstly we will start from the an introduction of fishes from S.S. Khanna book. So first of all, what is bioluminescence? Luminescence means light. Okay, so the fish which have capability to produce light, that shows the phenomenon of bioluminescence. Okay, mostly deep sea form fishes show this mechanism, which carried the luminescent organs which called the photophores. Photo means light. Okay, so these organs are derived from epidermis. We have many examples of fishes which shows the bioluminescence. The number and location of bioluminescent organs varied. For example, there are examples like we have scopulus and halosauropsis. The luminescent organs are located from head to tail. Okay, so in this scopulus and halosauropsis, the luminescent organs arranged in one or two rows extending from the head to tail. And in some fishes like opostomias, the luminescent organs are located in limited parts. For example, in opostomias they are arranged in transfer bands on the body. Okay, so there are large number of photophores are also present on the pectoral and dorsal fins as in insular fish, and there are very large number of photophores in the toadfish, and they are also present along the lateral line of the toadfish. Okay, the fishes with luminescent organs are worldwide distribution. It shows the cosmopolitan distribution, that is worldwide distribution, and majority of them are bathypelagic, living at the medium depth. They are approximate 500 to 500 meters and sometimes migrate to the surface at the night. Okay, so there are some examples with structures. Scopulus and halosauropsis, there are the luminescent organs located from head to tail. Okay, so next we have the light producing organs are found among the insular fish. We studied here also insular fish. There are a large photophores. Okay, so in insular fish there are present of bioluminescent organs, and insular fish order have the cerotoida, and other example latran fish, and their order is the stomatoida, and in scopulate. So we should learn scientific name of examples, so it impress more as comparison to common name. And when you write the scientific name of any fish, any organism, the first letter will be of the species will largest and largest in a capital, and the genus there will be smallest. Okay, so and it should be underlined. Luminescent organs are two types. So some fishes are self-luminescence. Okay, so but in others light is produced by symbiotic bacteria. So there are two types. First is self-luminescence, another by the symbiotic bacteria. Structurally, these photophores consist of large number of glandular tubules. These glandular tubules secrete the luminescence bacteria. For example, in Malacocephalus and the Coelurhinchus. Okay, this is the Malacocephalus. Okay, we will study later on. So in Malacocephalus and Coelurhinchus, the glands open by duct on the ventral surface of the fish. So a little in front of the anus. The gland is richly supplied by blood capillaries. In Malacocephalus and Coelurhinchus, there is a rich supply of blood. Other examples, photoplavron and anomalopes. Okay, in this it possess elongated luminescent organs. Okay, like this, elongated luminescent organs below the eye, and consisting of numerous long and parallel glandular tubules with rich blood supply. And this another anomalopes, there is a light is cut off by the moving at the light organs downward. In this fish, the light will cut from the downward by the means of hinge. Like we opens the door and close. There is a hinge present, and hinge joint in our legs and arms. Okay. So anomalopes, the light is cut off by moving the light organs downward by means of hinge, so that it comes into the contact with black pigmented tissue. When it downward, it contact with black pigmented tissue, and in photoblaphron, a fold of black tissue can be drawn up like an eyelid, eye open and close. Okay, so over the organ, thus cutting off the light. For in anomalopes, for example, anomalopes fish comes downward, so it decrease the luminescent function of fish. Okay, so there will be black pigmentation, and in photoblaphron there is a like a fold of black tissue like a eyelid. As we close our eye, so we saw the black pigmentation. Similarly, in photoblaphron there is a fold of black tissue can be drawn up like an eyelid over the organs that cut off the light. Next we have self-luminescent photophores. So self-luminescence means self, okay, without any help. So self-luminescent photophores are found in some elasmo branch, for example Sucilidae and Trophonidae, and in telos belonging to the families Tomitidae and Myctophidae. So these are the fishes which shows the self-luminescence characteristics of bioluminescence. Okay, so the suborbital organs of the Pachystomias microdon so is quite complex in structure. It is a cup-like, and in its wall is composed of several concentric layers. So you can see this. So this is the structure of Pachystomias, which consists of the concentric layers like refractor layer of spicules. This is gland, and this is the structure of the Pachystomias, which shows the suborbital photophores of the Pachystomias. This is the Malacocephalus laevis. It shows the luminescence characteristics. Next we have some chemicals like the extract from the luminescent gland of fishes like epagon and the perianthus. It consists of two enzymes and chemical. Enzyme is the luciferase and luciferin chemical. When this both combine, it produces the light. So how they produce? The luciferin from both of the species are identical and resemble closely with that obtained from the crustacean Cypritina. So how it produces? We all know that luciferin is an indole derivative. It consists the tryptamine, arginine and isoleucine, and this luciferin when mixed with enzyme luciferase, so luciferin is oxidized to oxyluciferin and emit the light. So this is the process in apogon and paraprianthus. So I repeat, luciferin derived from the indole derivative. It consists of three, that is the tryptamine, arginine and isoleucine. So then this luciferin is combined with luciferase enzyme. So when this both are combined, luciferin is oxidized to oxyluciferin, okay, and then emission of light in the apogon and the paraprianthus. Okay, next we have control and regulation of photophores. So the light producing organs are directly under the control of two systems, that is the nervous system and endocrine system. Okay, so the photopores of fishes are innervated and luminescence is undoubtedly under the nervous control. So some endocrine glands like suprarenal and other glands of the endocrines also activate them. Okay, so there are some examples which use the mechanical method for concealing the light organs or changing their brightness. So there are some examples like photostomias, anemalopes and photoblephron. It consists of membranous black curtain lies along the ventral edge of the organ, and emission of light takes place by the control of nervous system and endocrine system. So it is given in short. I will explain from the Pandey and Shukla book in detail. So next we have use and significance of light. Like we have a torch, we use torch for the detection of anything, and we can visualize things easily. Okay, similarly here light bioluminescence light help them to search the prey in the dark waters. Okay, like envelopes use this phenomenon to search the prey in the dark waters by the luminescent organs. Next are the significance. It attracts the prey. So light are attracted to the prey near the mouth of fishes, so they can eat them or feed on them. Next we have these luminescent organs act as a defensive organ in some species. For example, a sudden flash of light from a large luminescent organs may dazzle or confuse the enemy, and fish escape easily from the enemies. Next is it can be warning signals for the predatory animals like porichthes. So these fishes possess a toxin spine and avoided by the predators. So these porichthes produce the some toxin which is harmful for their predators. So this is the warning signals for some predators. Next is the patterns of photophores on the body is useful in the recognized individuals of the same species. Like same species can be easily identified. So it have basically 5 advantages, or we can say 5 significance. It helps in search, attract, defensive, warning signals, and recognize the individual of same species. Okay, so we done the bioluminescence from an introduction to fishes. Now we will do it from the Pandey and Shukla book. We will not do in detail because we already done. We will do here most topic which are not included in the SS Khanna book. Okay, so here what are the light organs we already done. So location already done. So this is the important families of light producing fishes. We can add this. Okay, so angelerfish, latronfish. So these are the families you can add or skip because we also done in SS Khanna book. So next we have categories, types we already done, self-luminescence. Okay, so in this we will do the mechanism of light from the luminescent organ. This is important and this is not present in the SS Khanna book. So the extract from the luminescent glands of several fishes like apogon and paraperianthus consist of luciferin and luciferase enzyme. This is also present in SS Khanna book but in short. So thereby we are doing from the Pandey and Shukla book. So in the apogon and perianthus there is a one chemical and enzyme. Chemical is luciferin and enzyme is luciferase. So when both these compound combine, it emits the light. Okay, in the presence of cation sodium, as you see that there is a L is luciferin and Hb is a photon. Okay, so when luciferin combine with luciferase and in the presence of sodium, it produces photons, that is the light. So what is the luciferin? We already discussed. It is the indole derivative and it consists of 3 compounds, that is the tryptamine, arginine and isoleucine. So when this mixed with enzyme luciferase, luciferin is oxidized to the oxyluciferin and finally it emits the light. Okay, so the luminescence in fishes is generally blue or blue green in color. So this is not added in SS Karna book. So however, it may be green or green yellow or blue white. Luciferin. What is the luciferin? We already discussed. It consists of three compounds, that is the tryptamine, arginine and isoleucine. It combined with luciferase oxidized. This luciferin oxidized to oxyluciferin and emits the light green, blue or blue green color of light. So this is the structure of luciferin that was given by Kishi in 1966. Okay, next is the regulation we already done under the control of nervous system and endocrine system. Okay, so there are some examples, we already done it. Next are the structure of certain light producing organs in certain fishes, like this. Okay, so we can draw these diagrams only, no need to written these. We can write it in short because in exams we should attempt our question according to time, so we can write in short. Okay, about these certain fishes. Next we have biological significance. We already done from SS Khanna book by its uses, but some uses which are not present in SS Khanna we will include from this. Search the prey we already done. Also dazzle species, yes, and prey attract the prey already done. And it has been reported that luminescence in polychaetes serve as a warning signal. The patterns of photophores on the fish body is useful. So these all the 5 significance of bioluminescent fishes is present in SS Khanna book. Next is the 6th significance. Fish which swim in the large shoals are able to keep together by recognizing the patterns of photophores. So sometimes these luminescent organs differ in size or patterns in the two sexes. So this is the useful for the recognition marks. So fifth significance was the recognition individual of same species, but in sixth we studied the recognition of marks. Okay, so these luminescent fishes belonging to this family, that is the Alipocephalidae, this produces the glowing spark which confuse the predators for a shot and allow two fish to escape. So the fish can easily escape from the predator, and luminescent also aid in the sexual recognition. We already done in 6, so we should add this 2 point, that is the recognition of marks. And some fishes of the family Alipocephalidae produce the glowing spark which confuse the predators. Okay, so this is all about the bioluminescent organs. If you have any query, you can simply comment in comment section. I hope this video will helpful for you. Thank you.